KCET 2025 aspirants, welcome back to KCET Master Stroke series, and today we are going to do one shot for the chapter Biomolecules. Okay, now many of the students who have generally taken computer science in their PU2 find this chapter very difficult, okay, and they generally try to avoid this chapter. But let me tell you, this chapter is a very scoring, low input, high output type chapter where you just have to remember certain facts and you will be able to score full on this particular chapter. Okay, so I'll be telling you all the important things that you need to remember, all the important concepts, terms, diagrams that you should be knowing to ace this chapter. Okay, so please watch this complete video, and those students who are afraid of this chapter because they have taken computer science, please let me know in the comment section. Okay, so let's jump to the first. So I'll be telling you what exactly we are going to study in this particular session. So see, biomolecules generally means what? Those molecules which help us to survive, okay, which are very much important for our growth, for our development, for our metabolism, everything. So these are the main constituent which helps us to survive. Now how many biomolecules are there? There are 5 major type of biomolecules: carbohydrates, proteins, vitamins, then you have hormones and nucleic acids. Okay, so did I miss? Yeah, enzymes is also there, but enzyme is a subclass of proteins. So I can say majorly carbohydrates, proteins, vitamins, nucleic acids and hormones. Okay, so now coming to the first thing, that is carbohydrates. Okay, weightage you can see 3 through 4 questions every year we get, but yes, again this year it will be more, 4 to 5 questions you can expect. Okay, so under carbohydrates, what all things we are going to study? First, what is classification of carbohydrates? How are the carbohydrates divided? Then what are monosaccharides, what are disaccharides, what are polysaccharides? So under this also, in monosaccharides we have about glucose. So in monosaccharides we will study majorly about glucose. In disaccharides we have you can see sucrose, maltose and lactose, we will study about them. And in then polysaccharides you can see we have for starch, cellulose and glycogen. So in monosaccharide we have 2 monosaccharides which we are studying. Glucose will be in much detail, and after glucose we will be studying about fructose. Okay, in disaccharides we have sucrose, lactose and maltose. Okay, and polysaccharides we have 3 polysaccharides again. One is your starch, one is your glucagon, and another is cellulose. Okay, so what are these monodiet polysaccharides? We will discuss that also. Okay, so first let us talk about classification of carbohydrates. Okay, so now you can see, yes, so on the basis of products of hydrolysis, see, when you break a carbohydrate, okay, it gives you smaller compound. So just imagine that carbohydrate is a polymer of saccharides or a polymer of one monomer units. So on hydrolysis, okay, if a carbohydrate is there and if you break it and if you are getting 2 units from that, then we call it disaccharide. Okay, so if you get only 1 unit, then it is called monosaccharide. So based on the product of hydrolysis we have monosaccharides, oligosaccharides and polysaccharides. So what do you mean by this? See, when one carbohydrate, okay, so these are small, suppose monomer units, okay, these monomer units combine to give you carbohydrates. Okay, so but if any carbohydrate is there which cannot be broken further, it is the monomer unit itself, that is called monosaccharide. Okay, oligosaccharide means when you break a big carbohydrate, if you get between 2 to 10, okay, 2 to 10 monosaccharides if you get, okay, between 2 to 10 monosaccharides if you get, then it is called oligosaccharides. Now polysaccharides means if you get more than 10, more than 10 monosaccharides if you get, then we call it as polysaccharides. Okay, now oligosaccharides, if you get 2 units then it is called disaccharides, if you get 3 units it is called trisaccharide, 4 units is called tetrasaccharides. Like this the classification is there. Now based on reducing nature, what we have, one second, yes, so based on reducing nature we have reducing sugars and non-reducing sugars. So if that saccharide, okay, is able to react with Tollens reagent, if they are able to react with Yes, if they are able to react with Tollens reagent, okay, then we call it as reducing sugars. If they are able to react with Tollens reagent and Fellings reagent. If they do not react, then these are called as non-reducing sugars. Okay. Next, moving forward, we have monosaccharides. So under monosaccharides, again these can be classified further. Okay. So now you can see, I told you monosaccharide is one unit. Okay. In that unit there can be many number of carbon atoms, right? So at least 3 carbon is minimum, because we need at least 3 carbon for having one monosaccharide. Okay. So triose it is called if it is 3 carbon. If it is 4 carbon, tetrose. 5 carbon pentose, 6 carbon hexose, 7 carbon heptose. So generally we will be dealing with this lucky number 6. Okay. Now based on the functional group, so this monomer unit, okay, it has how many carbons? That is what it can be distinguished like this. Now which functional group it has? So generally the carbohydrates have 2 functional groups, either aldehydes or ketones. Okay. So if they have aldehydes functional group, then we call it aldose. If they have ketone functional group, we will call them as ketose. Okay. Next I have monosaccharide. So first monosaccharides, I told you there will be 2 monosaccharides which we are going to study in detail. That is your glucose and fructose. So first one is your glucose, and how is it prepared? So glucose is prepared from sucrose. So what is sucrose here? Sucrose is a disaccharide. Okay. So when I break sucrose, one of the unit is glucose. Okay. So that is what you can see here. Yes. So you can see this is sucrose. If you hydrolyze it, if you break it, you get 2 things, right? You see the formula of both of them are same, but one of them is glucose, okay, and one of them is fructose. Clear? And both of these are monosaccharides. Okay. So that is one way how we can synthesize this one. Another method is from starch. Here starch is a polysaccharide, means a very big compound. Okay. So if I break a very big compound into with water hydrolysis, you will get n number of glucose units. Okay. So this is a commercial method of preparing glucose. This is clear? Okay. Now we will go to how to determine the structure of glucose. Okay. So the molecular formula of glucose is C6H12O6. Okay, we all know the molecular formula of glucose, but what exactly is the structure of glucose? To determine that, many reactions were conducted. Okay, and these reactions tells us the structure of glucose. So the first reaction that was done was reaction with HI. So if I do the glucose structure, I can just write like this, CHO. So glucose is an aldose. Okay, so the functional group here is aldose, and the number of carbon is 6, so it is a hexose as well. So the structure is like this. You can just imagine like this. Okay, this is glucose, 6 carbons are there. I have a aldehyde functional group, and this is what I can think about the structure. Now if you react this one with HI, okay, we get that is structure called as N-hexane. Okay, how is this structure? CH3, CH2, 4CH3. Okay, now what does this tell you? That glucose, all the 6 carbons are in one chain. Okay, there is no branching in that. So what I can conclude from here, all carbon atoms are in one chain. Clear? Now moving forward, what we have? Reaction with NH2OH or HCN. So now what will happen? This is CHO. Okay, you have CH2 whole 4 and you have CH2OH. Now if you react with HCN, so what will get? A cyanohydrin will form here, COH, CN, right? CH2 whole 4, CH2OH. Okay, if you do with NH2OH, hydroxyme will form. We all know aldehydes ketones reaction. So what conclusion does this one gives us? So this conclusion tells us that there is one carbonyl group. We don't know whether it is aldehyde or ketone. Okay, a carbonyl group is present that can be either aldehydes or ketones. Now see, you might be thinking that ma'am has already told that it is aldose, then why this confusion is there? See, that is after we have derived the structure, we know that it is an aldehyde, but before we just knew the formula. When scientists discovered this one, see, okay, we were just having this C6H12O6. We don't know anything about this. So we have done so many reactions to determine the structure of this one. Okay. So once this was done, okay, once this reaction was done, that glucose molecule was taken and reacted with HI and we got N-hexane. We got the first conclusion that all the 6 carbons is one chain. Next what we did, as we reacted with NH2 and HCN, okay, then we know, okay, it is giving reaction with NH2OH, it is reacting with HCN, then definitely it will have one carbonyl compound. It can be either aldehydes or ketones. Now what we did, we reacted with bromine water. So what does this bromine water does? Bromine water is a mild oxidizing agent. Okay, so what will happen if it is a mild oxidizing agent? What can it will do? See, aldehydes can be so we want to find out whether it is aldehyde or ketone. So if I take an oxidizing agent, if it is ketone, there will be no reaction, but if it is an aldehyde, it will get converted to acid. Okay, so what was there? We have this formula only, right? Okay, now if I treat it with this one, bromine water, so what I found one COOH group was formed there. So now what will I conclude? That I know one of the carbonyl compound is present, but on a mild oxidizing agent, if that carbonyl compound is getting oxidized to acid group, then definitely I can conclude here that aldehyde group is present. Right. So till now what all things we have concluded? There is a long carbon chain of 6 carbons. Next what we concluded? There is a carbonyl compound, aldehyde or ketone. Third, we confirmed that it is an aldehyde group. Okay. Next what is there? Okay, now you can imagine that there are C6H12O6, 6 oxygen is there. One will be in aldehyde, we know. What about the 5 OH groups? What about the 5 oxygen groups that is present? So in what form are they present? So to find out that, what we did, we have taken the same compound, okay, and we reacted with this one, that is your acetic anhydride. Okay, now if we have any alcohol group here, okay, then what will happen? Alcohol with acetic anhydride will give you esters. So now we found that a pentaacetate group was formed, means 5 ester groups were formed. So that 5 ester groups, what did conclusion we get from here? That in this, 1 oxygen is involved in aldehyde group, we understood. The other 5 groups, 5 oxygen are involved in 5 OH groups. Okay, next we did reaction with HNO3. Now to understand whether these OH groups, okay, are primary OH group or secondary OH group or tertiary OH group. So what is it? It is a oxidizing agent, strong oxidizing agent. In this case what will happen? If primary OH is there, it will get converted to acid. Okay, so now I know that there is one aldehyde group and there are 5 OH groups. Okay, now when I treated with HNO3, I got 2 COOH groups are present now. Okay, here something I don't know, but in the result I found 2 COOH group is present. Now how is that possible? One COOH group we know because of the oxidation of the aldehyde group. Okay, I have one aldehyde group that will get oxidized to give you one of the acid group. Now I am getting another acid group, and that is possible only if one primary hydrogen is present, primary OH group is present. So that confirms the presence of one primary OH, and out of 5, 1 is primary and 4 secondary OH. Okay, so that is how finally, after doing all these reactions, we came to the conclusion that this is the structure of glucose, where we have 4 secondary OH, we have 1 primary OH, and we have 1 aldehyde group, and all the 6 carbons are in one chain. Clear? Okay, now what we have? So this is the open chain structure of glucose, but it had some limitations. As you can see, it does not react with Skiff's reagent, it does not react with NH2OH, okay, and it exists in the 2 form. So the major thing you can see here is it exists in 2 form. There is 1 alpha form and 1 beta form. So how did this alpha and beta form came? So that is because what happened here is, it was suggested that this glucose can also exist in a cyclic form. It can exist in a straight chain form that we have already seen, but it also exists in a cyclic form. And what is that cyclic form of glucose? That is how this one OH combines with CO double bond O group. Okay, there is a reaction here, a hemiacetal is formed. Okay, so that is why this is the structure of the cyclic ring. And the Haworth, it is not looking like a cyclic ring, right? So to represent it in the cyclic structure, these are the two structures of glucose. Okay, so these are the two cyclic structures of glucose. Two structures, why it is there? Because it exists in alpha and beta form. So if OH is towards the down, okay, this one carbon OH is towards the down, then it is called alpha. If the OH is towards the up, it is called beta. Now you see the configuration is same in all the carbons, but the configuration is different only in this one particular carbon. Okay, such type of isomers where the configuration is different on any one carbon, okay, that is called as your anomers. Okay, such type of isomers where the configuration is different at any one position, that is called as anomers. Okay, now moving towards the next one. Okay, yes, so as I told you, there we have one more monosaccharide, that is your fructose. So it is also very similar, but here we have a ketone group in fructose. Okay, so it also has cyclic structure, so you can see, but it is a 5-membered ring. Glucose is a 6-membered ring. Okay, so it is similar to the structure furan, and glucose is similar to the structure pyran. Okay, now if you see here, this is OH is towards the down, so it is alpha. Here OH is towards the up, so it is beta. These two are also anomers. These type of isomers are also called as anomers. Clear? Next, moving forward. Okay, now we will go to disaccharides. So for every disaccharide, you should be knowing some important things. What are the things you should be knowing? I'll make a table. So you have to look this. So you make a table and you have to fill the table after seeing these slides. Okay, so what is there? The first thing you have to make is monomer unit. Okay, you should make like a table like this. Okay, I really want all of you to read this in the form of a table. We have 3 disaccharides. Okay, so first one is sucrose. Okay, next one is your maltose. Okay, next one is your lactose. Okay, so first column should be your monomer units. Now disaccharides means there will be 2 units. Okay, so what are the 2 units present in this disaccharide? That should be your first table. Okay, next one you have to put linkage. Okay, so the two groups should be connected, right? So which carbon to which carbon there is connection, that should be written in the second one. Very, very important about the linkage part. Now third is whether they are reducing or non-reducing. Okay, so whether they are reducing or non-reducing should be the third one. Okay, so this is how you should be studying all the 3 disaccharides by making a table. Very easily in a compact place you can write all the properties, and that will help you to retain better also. So coming to the first thing, that is your sucrose. So sucrose is made up of D-glucose and D-fructose. Okay, so this should be written in this monomer units. Next, what is the connection? You can see it is connected between D-glucose, C1 of D-glucose and C2 of fructose. Okay, so you can see alpha D-glucose and beta D-fructose connection is there. C1 of alpha D-glucose and C2 of beta D-fructose. Next, it is a non-reducing sugar. Okay, next, fourth point, you can draw the diagrams. Okay, you can also draw the diagrams after this. Clear? You can see here. Now what is this bond between 2 disaccharides called as? It is a glycosidic linkage. Okay, and you should remember this glycosidic linkage is a type of ether linkage. Okay, it is an ether group. Clear? So that is what you have to know about each of the disaccharide. Next, moving forward, we have for this particular sucrose, there is something called as inversion. Now see, sucrose is generally levorotatory, means dextrorotatory, means it will rotate the plane polarized light in anti-clockwise direction. clockwise direction. It is dextrorotatory, right? So it will rotate in plane polarized light in clockwise direction. Now you have put some acidic water to glucose solution, sucrose solution. So what will happen? Sucrose will hydrolyze, and if it is hydrolyzing, what it will produce? Glucose and fructose. Now in glucose and fructose, what happens? Glucose is actually dextrorotatory, but fructose is levorotatory, and fructose is more levorotatory than glucose. So overall what happens? Finally, when sucrose undergoes hydrolysis, the solution becomes levorotatory. Okay, it started with dextrorotatory, it undergoed hydrolysis, and after that we found that the solution has become levorotatory. This process is called as inversion of sugar. Okay, so that is because of the levorotatory of fructose and dextrorotatory of glucose. Okay, next we'll move to next disaccharide, that is maltose. So in maltose, the monosaccharide units are your alpha D glucose units, and the connection is between C1 and C4, and it is a reducing sugar. Okay, next we have that is your lactose. So lactose is made up of beta D galactose and alpha D glucose. Here also the connection is from C1 to C4, and this is also a reducing sugar. Okay, next we'll go to polysaccharides. Okay, so polysaccharides also we have three polysaccharides. One is your cellulose, starch and glucosan. So starch is something where we have little more to study, other two very simple things. So in first thing, that is starch. Okay, so it is a polymer of alpha glucose units. Okay, and it is two types, amylose and amylopectin. In amylose, what happens? It is water soluble, this is water insoluble. It is present in less amount, it is present in more amount. It is an unbranched chain, means it is a straight chain, but this is a branch chain. So if it is a straight chain, there are monomer units. What are the monomer units? Alpha D glucose units. So how are they connected? They are connected between C1 to C4. Now if there is branching, it is connected between C1 to C6. Okay, that is the difference. Now we'll go to the next one, that is your next polysaccharide, which is, so you can see the diagram of amylose and amylopectin. It is a straight chain, and this is a straight chain branch chains, then again branch straight will go on. Okay, now moving to the next one, that is cellulose. So cellulose is a polymer of beta D glucose units. Okay, it is not a polymer of alpha D, it is a polymer of beta D glucose units. Okay, now here what is the linkage? Same C1 and C4, and it is the use, you know, cellulose is found in the cell wall of the plants. And next, if I see glucagon or glycogen, from that time I am saying glucosan, sorry, it is glycosan. Okay, so glycosan is present in the animal body as starch. Okay, now it is also having similar structure to amylopectin. Okay, it has similar structure to amylopectin. Now what was the structure of amylopectin? It is straight branch like this, right? So that is what is happening here. It is found in yeast and fungi also. Okay. Okay, now there is some question here, how many monosaccharides are obtained in the hydrolysis of sucrose? So sucrose, it is a disaccharide, so you will get 2 monosaccharide units. Okay, next, which of the following does not reduce the Benedict solution? So to reduce Benedict solution, you should have a reducing sugar, right? But we know that glucose is not a reducing sugar, so it will not react with Benedict's solution. Not glucose, sorry, sucrose. Next, the pair of compounds showing positive tolerance reagent is. So positive tolerance reagent will be for sucrose is not there, right? It is a non-reducing. Glucose and fructose, option number C. Fine. Next, we will study the next biomolecule, that is proteins and enzymes. Okay, very easy, small thing, small topic. See, in proteins, what all things we have to study? What is the amino acid, classification, structure and denaturation. Okay, now generally see, carbohydrate is made up of saccharides. Proteins are made up of amino acids. Okay, so let us see what is that. So amino acid structure is like this. You have one carbon, one hydrogen, you have one COOH group attached, you have one NH2 group attached, and then there is one R group. Now this R group can be anything. Based on this R group, we have total 20 amino acids. Okay, total we have 20 amino acids. So you can see these all are the amino acids. Now what happens, this amino acids, you can see as the structure is like this. Okay, so you can find this is an acidic group, this is an base group, so this can donate H plus, this can accept H plus. Okay, so we find like this, the structure becomes like this, and this is called as a Zwitter ion structure. Why Zwitter ion? See, it is a neutral molecule, but it is a polar molecule as well, because we have one negative charge and one positive charge. So generally amino acids exist in the form of Zwitter ion. So what are amino acids? Amino acids are the constituents of proteins. Okay, proteins are big macromolecules, but they are made up of the monomer units called as amino acids. Now amino acids generally exist in the form of Zwitter ion. Next, what we have, the classification of amino acids. Okay, one second. Yes, so amino acids can be classified, okay, into acidic, basic or neutral. Okay, how do we do that, acidic, basic or neutral? So now generally what happens, acidic amino acids, we have seen that we have one C group, one H group, COOH group, NH2 group and R group. Now in this R group, if there are COH group also, so overall in the compound you can see more number of acidic groups will be there, then it is called acidic. If more number of NH2 amino groups are there in the R group, then it is called basic. If equal number is there, then it is called neutral. Next we have, based on synthesis, essential and non-essential amino acids. So essential amino acids are those amino acids which, okay, cannot be synthesized by body. Means we have to consume that from the diet. Non-essential are those which can be synthesized in the body. Okay, so there are total, these are the essential amino acids. I have a better list to show you. You can see here, valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, tryptophan and histidine. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 acids are there, 10 amino acids are there which are essential amino acids. You should be knowing the name of each of them. Okay, next, okay, so I have one second. Okay, I have I will say what it is, mnemonic to remember this one, but I actually will tell you. So you can have like this, okay, one second, you can have like this, PVT, okay, then I have TIM and then I have HIL. Okay, so P stands for phenylalanine. Okay, this is how I have remembered. Then V stands for valine. Okay, then T, you have two amino acids here, threonine and tryptophan. You can consider anything here, threonine. Okay, then you have tryptophan. Okay, then you have isoleucine here. Okay, then M, we have methionine. Okay, then HI stands for histidine. Okay, then I have leucine and I have leucine and lysine, right? Yes. Okay, so private, TIM, HIL, this is there, but I have missed A, right? So it is A, you can just put private, TIM, HIL, something where A can be added. Okay, so that is how I have remembered. You can also follow this, or you can make your own mnemonics. Okay, now coming to peptide bond. So as you see, disaccharide units, okay, they were connected by glycosidic linkage, which is a ether bond. Similarly, the amino acids, okay, amino acids are linked by a bond called as peptide bond. Okay, now in this peptide bond, what happens? Now see, if you have this, this is one of the amino acid, right? So here, from here, what will happen? Water will be lost. So one H from here and OH from here will be lost. So you will have CONH group. Okay, that is what is present, and this is called as a peptide bond. Now in this peptide bond, if two, so here two amino acids are joined, right? So that is called as dipeptide. So in a dipeptide, there is one peptide bond. So if I have a tripeptide, means I have 3 amino acids. So I will have 2 peptide bond, or I can say it is a, in tripeptide there will be 2 2 peptide bonds. In tetrapeptide, 4 amino acids will be there. There will be 3 peptide bonds. So that is what you should be knowing. So now what happens? These all are called as, these, suppose I have made 3, these are all called as polypeptides, like 2, 3 amino acids I have combined, 5, 10 I have combined. But when so many amino acids are combined and that unit molecular mass is higher than 10,000 atomic unit, then we call it as a protein. Okay, so that is always not correct. Okay, we have insulin, okay, it is a protein, but it has only 50 amino acids. But generally this is a rule, but not followed exactly always. Okay, next coming to the structure of protein. Okay, so the structure of protein can be understood in this way. So the protein structure is of 4 level. Okay, primary, secondary, tertiary, quaternary. Means if you open the structure of the protein, first what you will visualize is the quaternary structure. Then if you unfold it, you will go to tertiary structure. Then if you unfold it, you will go to the secondary structure, and at the end you will find the primary structure. Okay, now what is, first we will understand this one, then we will understand this one. Now what is primary structure? So just imagine that you have a thread and you have beads in that. Okay, you have a thread of beads and each bead there represents 1 amino acid. That is the primary amino acid. Okay, you have the linkage, when the amino acids are linked in a particular order for a particular protein, that is called as primary structure. Okay, now what happens? What happens in secondary structure? In secondary structure, this protein can coil or can change its shape. This one thread can change its structure. So if it is undergoing a coiling type, okay, then it is called alpha helix structure. So secondary structure can be either alpha helix, okay, where the protein is coiling around, okay, it is like, it is in this shape. Or else what happens, this is one thread, this is one, 2 threads are coming towards each other and they are forming some bonds between them, and we are getting some sheets like structure. Then that is called beta pleated sheet. Clear? Now in tertiary structure what happens? 2 coil came, suppose, okay, this is one coil, this is one coil. They came to each other and they got a spherical shape. Then that is called tertiary structure. Or if we have so many beta pleated sheets are there and they come together, okay, and they form a sheet of structure like this, or a thread like structure, then that is called fibrous and globular. So in tertiary structure, based on if it is alpha helix, then we get globular protein, okay, and if it is beta pleated sheets, then we get fibrous proteins. Okay, now if there is one globular protein here, one globular protein here, one globular protein is there, now all the globular proteins assigned together, okay, then we get quaternary structures. Okay, so generally the protein can be in the form of quaternary structure or can be in the form of tertiary structure. Okay, next if I show you what is the difference between fibrous and globular. So in this what happens, the polypeptide chains run parallel and are held together by hydrogen and sulfide bonds. So this is very important. What bonds are there? This is hydrogen and sulfide bonds, and they have a, here they have spherical shape. Now this is insoluble, this is soluble. Where it is found? Keratin and myosin, and this was found in insulin and albumins. Okay, now next is yes, primary structure I have already given you. Okay, so you can see in primary structure it is just the sequence of amino acids. In secondary structure you have alpha helix and beta sheeted. In alpha helix you have the regular folding of the polypeptide chain due to the hydrogen bonding. Okay, so regular folding is there. Okay, and there is a hydrogen bonding between the CO group and NH group. Okay, and in beta plate sheets what happened? They are laid side by side and are held together by intermolecular hydrogen bonds. Here what you have? Here also hydrogen bonds, here also we have hydrogen bonds. So you can see here this is one chain, right? So here there is a coiling between them. Okay, there is a coil and you can see CO group of 1 and NH group of 1 is forming bond with each other. Okay, now in this case what happens in beta pleated sheets? These are like this long chain arranges in such a way that it forms a sheet type structure. Okay, next we have here Yes, tertiary and quaternary structure. So in tertiary there is further folding and we get fibrous and globular, but how the tertiary structures are held? They are stabilized by hydrogen bonds, disulfide, van der Waals force and electrostatic force. This is very important and can be a potential MCQ question. Now coming to quaternary structures, the spatial arrangement of two or more polypeptide chains are referred to as quaternary structure. Example is hemoglobin here. Okay, so you can see a basic diagram. You can see that here the primary structure is there where only the amino acids are present. Now here you can see these amino acids are coiled, okay, and here they are arranged in a sheet. Now in tertiary structure what happens? Further coiling of this one takes place, and here this is the quaternary structure where further coiling of tertiary structure takes place. Okay, that is a okay. Now something called as denaturation of protein. Now protein can function efficiently if they have a particular temperature, a particular pH. Okay, now if you disturb, if you change the temperature, if you change the pH, then these hydrogen bonds and everything is disturbed. Okay, and the protein loses its biological activity. That is called as denaturation of protein. Okay, now when you denature, the tertiary structure will uncoil, you will get secondary, quaternary will uncoil to tertiary, tertiary will uncoil to secondary, secondary will uncoil to primary. So at the end of denaturation of protein, you will have your primary structure intact, but all other structures will be destroyed. Okay, this is also very important to know. Now examples of structure is what we have: coagulation of egg white and curding of milk. So generally what happens, in egg the albumin is present, right? It is present in this structure, but when you boil it, when you heat it, the temperature, so we get the protein gets denatured and we get in the, what I'll say, in the primary structure form. Okay, now if I move to enzymes, so enzymes are very simple. They are what? Biocatalysts. Enzymes are generally proteins and they are very specific. So every enzyme has one particular function and they mind their own business. Okay, so that is what they are, very very specific in their activities. Okay. Okay, so here is a question: glycosidic linkage involved in glucose units in amylose part of the starch. Amylose, C1 to C4, right? C1 C4 alpha linkage. Okay, next question: proteins are found to have different types of secondary structure like alpha helix and beta pleated structures. Alpha helix structures of proteins is stabilized by hydrogen bond. Even beta pleated is also stabilized by hydrogen bonds. Okay, so it's the same question. Okay, now we will move to the last part or the most important part, that is vitamins, nucleic acids and hormones. Okay, so in vitamins what are we going to study? Okay, in nucleic acid what are we going to study? Its chemical composition, structure and function. Okay, next in vitamins what we are going to study, I'll tell you: classification and functions. Okay, similarly in hormones also we are going to study classification and functions. Okay, fine. So now what are exactly vitamins? Vitamins are those nutrients or those things which are required in very less amount in our body. Okay, they are required in small amount and that is generally taken from the diet. Okay, vitamins are not produced in the body. So now vitamins are of two types. One is fat soluble and water soluble. Okay, so fat soluble vitamins are what? A, D, E and K. They are not soluble in water. So that is why these vitamins can be stored in liver and in adipose tissue. But water soluble vitamins are B and C. So what happens when we urinate or when we sweat? These vitamins get dissolved and they are excreted from our body. So it is very important to take vitamin B and C regularly from the diet. Okay, suppose A, D, E, K you have taken, it will be stored in the body, but B, C you have taken, it will again go out of your body. So regularly you should consume. Okay, next, this is the damn important, very very important table where you have the vitamins, their sources and their deficiency disease, and I cannot tell you how many times the questions have been asked from this particular table. Okay, so you should definitely study this one, you should remember it. This is something you would have studied in class 6, 7, 8, basic general knowledge. So please learn this table. And for vitamin you should be knowing their chemical names also. B1 is called as thiamine, B2 is called as riboflavin, B6 is pyridoxine. Okay, vitamin C is ascorbic acid, their deficiency diseases, this you have to remember. Okay, now coming to nucleic acids. 1 second. Yes, so nucleic acids, you all know RNA and DNA, we have studied many times, we have heard this, right? So nucleic acids are of two types. What is it? It is RNA and it is DNA. Okay, now a nucleic acid, okay, like as I told you, carbohydrates are the polymers of saccharide units and they are connected by which bond? They are connected by glycosidic ether linkage. Proteins are the polymers of amino acids and they are connected by peptide linkage. Similarly, nucleic acids are polymers of nucleotides. Okay, nucleic acids are the polymers of nucleotides, okay, and we'll talk about them in detail. So, okay, just let me show you what is there. Okay, now see. Every nucleic acid is composed of what? Nucleic acid is composed of nucleotide. Okay. Now this nucleotide is made up of three things. One pentose sugar, one phosphoric acid and one nitrogenous base. Okay. Every nucleotide is made up of three things. It should have a pentose sugar, it should have a phosphoric acid and it should have a nitrogenous base. Okay. Now the pentose sugar, if it is RNA, okay, so RNA will have, now see how is the composition, let me show you. See, suppose I have DNA, okay, DNA will be what? This is nucleotides, okay, this is nucleotides. Each of this nucleotide contains three things, okay. What is there inside this one? You have one sugar, pentose sugar, you have one phosphate group, okay, and you have one nitrogenous base. Clear? Now this, if it is a DNA, then this sugar is your deoxyribose sugar. Deoxyribose sugar. Now if it is a nitrogenous base, the nitrogenous base present in DNA is your adenine, guanine, thymine. So the nitrogenous bases present in DNA are adenine, guanine, thymine and cytosine, and phosphate group is present like this. Clear? Now if instead of DNA I have an RNA, okay, then RNA will also be made of nucleotides, right? Then what is the difference, ma'am? How can we differentiate between DNA and RNA? Both are made up of nucleotides, right? But yes, there is difference. Now see, each of these nucleotide is made up of again three things. What are they? Sugar, phosphate group, okay, and nitrogenous base. Now what is the sugar present here? Here the sugar present is deoxyribose, here the sugar present is ribose. That is how one way how we can differentiate. And what are the nitrogenous bases? Here adenine, guanine, thymine and cytosine. Here we have adenine, guanine, okay. There is no thymine here, we have uracil in presence of that, and you have cytosine, okay. So by this way we can differentiate between the DNA and the RNA. Okay, so basically both DNA and RNA is made up of nucleotide. Each nucleotide is made up of three things: sugar, phosphate group and a nitrogenous base. The sugar in DNA is different. The nitrogenous base in the DNA and the nitrogenous bases in RNA are different. Okay, so 1 second. Yes, so that is part clear. So I can say that in RNA, what sugar I am using? Deoxyribose. So you can see here, in this ribose sugar, this is the structure, okay. But in DNA, this is the sugar used where you have no O group, okay. This is a deoxy sugar group. Now similarly, if you see the nitrogenous bases, RNA has adenine, guanine, uracil and cytosine. Here we have adenine, guanine, thymine and cytosine, okay. Okay, next, what is something called as nucleoside? Now see, I told you a nucleotide is made up of three things: a nitrogenous base, a sugar group and a phosphate group. But if I have only nitrogenous base and sugar, if I just add these two, then we call it nucleoside, okay. To this nucleoside, if I add a phosphate group, it will become a nucleotide, okay. So that is just a subdivision between that. Okay, so now where is this bond formed, or where is the connection formed, you have to see. Okay, so now see, so this is your what? This is your pentose sugar. On the first carbon we have the nitrogenous base. Okay, so this pentose sugar and this nitrogenous base together forms your nucleoside. Now on the fifth carbon, okay, this fifth carbon, you can see we have the phosphate group attached. Okay, the phosphate group is attached to the fifth carbon. So I have first carbon the base, fifth carbon is your phosphate group. Okay, next what I have to show is the phosphodiester linkage. So two nucleotides, okay, will be joined to each other, then only you will get a big nucleic acid. So how do they join? They do join by phosphodiester linkage. Okay, now what is that? You see, so this is one nucleotide, this is one nucleotide. Okay, just understand the diagram, you don't have to remember anything. This is 3, 4, 5. So first carbon base is present, fifth carbon phosphate group is present, right? Yes. Now what happens? The same thing is present here, 1, 2, 3, 4 and 5. Now what happens? There is a there is a phosphodiester linkage. Okay, the phosphodiester linkage happens between what? You can see between the phosphate group of this fifth carbon. Okay, the phosphate group attached to the fifth carbon and this third carbon. Okay, the third carbon of the one group and the phosphate group attached to the fifth carbon of one group. So there is a bonding between 3 and 5. Okay, so that is called as your phosphodiester linkage, and we name it as 3' and 5'. Okay, so there is a bonding between 3' to 5' carbon atoms. Okay, clear? So that is about the nucleic acids. Okay, okay. Next what happens? This nucleic acids, suppose one nucleic acid is there, another nucleic acid is there. So how do DNA exist? DNA exists in the form of two nucleic acids. Okay, two nucleic acids coil around each other to give you DNA, whereas RNA exists in the single stranded form, DNA exists in the form of double stranded. Okay, so that is there. Now here there is something about complementary. So let me see if I have the diagram. Yes, this is the structure of DNA. You can say this is one nucleic acid. Okay, this is one nucleic acid and this is another nucleic acid. Now there is hydrogen. So what are these bonds present? These are the nitrogenous bases which forms bond between them. Okay, so now how the bond is formed? So always remember, adenine forms hydrogen bonds with thymine. Okay, so A is there means, suppose this is one strand, this is another strand. If here A is there, then this has to be T. Okay, hydrogen bond will be present between A and T. Similarly, G and C will form bond. ATGC will be complementary to each other. Okay. Okay. Next, okay, let me explain this one in little bit detail because this is from where questions can be asked. Suppose you have one DNA strand. Okay, two DNA strands will be there. Okay, two nucleic acids will always be together to give you a DNA strand. Okay, so now here suppose the nitrogenous base nucleotides are present. Okay, these are the nucleotides. Okay, and in each nucleotide there will be one pentose sugar. Okay, suppose here there is a pentose sugar, there is a phosphate group here, and there is a nitrogenous base here. Right, so this nitrogenous base is suppose A, adenine, then here the nitrogenous base has to be T. Suppose here it is G, then here it has to be C. So if I know the nitrogenous base order of one of the chain, suppose it is A, G, C, C, T, A, C, G, A, G, something is like this, then this is of the first strand, then can we guess for the second strand? Yes, definitely. Why? Because if it is A, it has to be T, G means C, C means G, C means G, T means A means T, C means G, G means C, A again T, G, C. So by looking at one strand nucleotide bases, you can easily guess the other one. So that is called complementary strands. Okay, the nitrogenous or the DNA strands are complementary to each other. Okay, now we have the nucleic acids function. It stores the genetic information responsible for protein synthesis. Now we will come to something which is very important and most ignored part from this chapter. Most of you do not study this part seriously, but questions are repeatedly asked from here. And I am telling you, the way I have given this table, nowhere it is mentioned, no book can give you. So please make sure that you learn this table, you write down this table, take a screenshot of this table and then study. Okay. So first, what is hormones? Hormones are your intracellular messengers. okay, intracellular messengers. Okay, next we have, they are selected by endocrine glands, okay, and they are put directly into the bloodstream. Okay, so these are to pass messages between two cells, okay, this or between the inside the cell. Okay, so we need these hormones, okay, and what they do, they are secreted by endocrine glands and they are directly poured into the bloodstream. Okay, like how we put garbage directly to the river stream, right? Similarly, these endocrine glands are present, they directly put the hormones into the bloodstream. I know it's a very bad example, but just to understand. Okay, clear? So pour directly means you might be thinking then how to put indirectly, right? So it doesn't have any cover. Okay, it doesn't have anything covering or any separation. The hormones are released by the endocrine gland and directly it mixes with the bloodstream. Okay, okay. Now hormones are also classified under three types according to the book that is given to us, NCERT book. It is polypeptides, it is amino acid derivatives, and it is steroids. Okay, three classifications are there. Now what hormones or examples are there under polypeptides? You have insulin, endomorphins, and glucagons. Okay, polypeptides, insulin, endomorphins, and glucagons. In amino acid derivatives, there we have epinephrine, norepinephrine, and thyroxine. Three there, three here. Steroids, we have estrogens and androgens. Okay, remember polypeptides, we have insulin, glucagons, and what was it? Endomorphin. Then amino acid derivatives, here we have epinephrine, norepinephrine, and thyroxine. Then in steroids we have estrogens and androgens. Okay, okay. Now we will see their functions. Yes, so you remember this one, glucagon and insulin, these were of what type? These were of your polypeptides type, right? So what is their function? Glucagon increases the glucose level, insulin decreases the glucose sugar level. So both of them together maintain the sugar level in the blood. Next we have epinephrine and norepinephrine. So this is what this was your amino acid derivative, right? So they respond or they maintain the responses to external stimuli. Next we have another amino acid derivative, that was thyroxine. Now what does thyroxine do? It is produced by thyroid gland, endocrine gland that produces this one is your thyroid gland. Next it is the iodinated derivative of amino acid tyrosine. So tyrosine is one of the amino acid. In that something is replaced, one hydrogen is replaced with iodine and we get thyroxine. Now if it is present in low amount in our body, then it causes hypothyroidism, whose symptoms are lethargy and obesity, and if it is in high concentration, it creates hyperthyroidism, which leads to enlargement of the thyroid gland. Okay, next yes, okay, next is your steroid hormones. Okay, the third category, right? Steroid hormones are again of two types, one which is produced by adrenal cortex and the one which is produced by gonads. Okay, now what all are produced by adrenal cortex? You have glucocorticoids, mineral corticoids. These two are produced from adrenal cortex. What do? They are released by adrenal cortex, and what is the function? They control the carbohydrate metabolism, inflammatory reactions, and involved in the reactions to stress. Now mineral corticoids, these are also synthesized by adrenal cortex. Their function is to control the level of secretion of water and salt by the kidney. Okay, now we have something called as addition disease. So this happens if your adrenal cortex is not functioning properly. If adrenal cortex will not function properly, then glucocorticoids and mineral corticoids will not be synthesized properly. If they are not synthesized properly, we will have the certain diseases, that is your hypoglycemia, weakness, and increased susceptibility to stress. You can see here that glucocorticoids was responsible for maintaining stress. If your glucocorticoids is not synthesized properly, then definitely you will be more stress, your stress will not be managed properly. Okay, next we have here Next what we have, yes, so next one is those hormones that is synthesized by gonads. Okay, so under gonads you have first one is testosterone, estradiol and progesterone. So testosterone is your major sex hormone that is produced in males and it is responsible for the secondary male characteristics. Similarly, estradiol is what female sex hormone. Okay, so it is responsible for the secondary female characteristics like your menstruation, and controlling the menstrual cycle, the development, body growth, all the secondary features are there which is responsible, and that is done by estradiol. Next we have progesterone, and that is also a sex hormone and it is used for preparing the uterus for the implantation of fertilized egg. Okay, so these are the functions of the hormones. Now let us see one question here. What makes up the primary structure of protein? So primary structure of protein, we all know it is made up of amino acids. Okay, so okay, next question. DNA fingerprinting is now used in forensic laboratories to identify the racial group, to determine the paternity. So all of this, so answer is option number D. Okay, now we have next question. Which of the following is not present in nucleotide? Okay, so if you see here, adenine, guanine, cytosine is there. Tyrosine is a amino acid. It is not a nitrogenous base, so it is not present. Option number D. Okay. Next question, yes, that's all students. We have done this one shot for biomolecules, and I hope that you would like make all the important notes, whatever structure. So basically carbohydrates is the major portion from where you can expect the more number of questions. So make sure that you remember all the examples that is given. Okay, so that's all, and see you soon in the next class, and do not forget to subscribe the channel. Thank you for watching.